Hi, it's Dr. LeBlanc again. The title of this video capture is RNA. Many investigators in molecular biology consider the possibility that life on Earth evolved first as RNA, just RNA, because RNA can be both a genetic material and an enzyme and a structural molecule. In this video capture, we're going to learn about ribose and its chemical properties. We're going to learn about the bases found in RNA and features of RNA that contribute to its different structures and functions in the cell. RNA is typically copied from DNA in the process of transcription. In RNA synthesis, only one of the two strands of the DNA is used as a template, and the primary structure of an RNA strand is much like that of a DNA strand. It has a 5' end and a 3' end. It has phosphodiester linkage on one side, and the bases protrude into the middle of the molecule. RNA strands are typically several hundred to several thousand nucleotides in length, although there are some very, very important small RNAs in the cell that contribute to gene regulation and embryonic development. So, the last video capture lecture, we talked about deoxyribose. Ribose has an oxygen attached to the 2' carbon, right here. And remember that the carbons in the ribose sugar are numbered from the first point of attachment of the base, and they're numbered as 1' to distinguish the carbons of the ribose from the carbons of the base. Our 2' carbon is where the hydroxy is attached for ribose. Our 3' end and our 5' end carbons are the ones that are involved in a phosphodiester linkage. And remember, the 5' carbon dangles off from the ribose structure itself. Here are our dancing carbons. Okay, so if we were going to compare the two structures of deoxyribose and ribose, they both have a hydroxyl on their 3' end when it's the very end of the molecule and it's not involved in a phosphodiester linkage, but it's the 2' carbon that distinguishes the two sugars. In deoxyribose, there's nothing here but a hydrogen atom, and ribose, of course, has the hydroxyl group here. That hydroxyl, by the way, makes it rather an unstable molecule, and we saw this structure in the last video capture as well, where we have adenosine, which is just the base and the sugar, adenosine monophosphate, a single phosphate attached, 2 phosphates, adenosine diphosphate, and our favorite molecule, ATP. These, of course, are the bases that you could have for different nucleic acids, the unmodified bases. There are actually a whole slew of other bases that can be attached or modified from the existing bases. Adenine, guanine, cytosine, and thymine are the 4 bases of DNA, and adenine, guanine, cytosine, and uracil are the 4 bases in RNA. Uracil takes the place of thymine. The difference between thymine and uracil is just this methyl group here versus a hydrogen atom here. Our phosphodiester linkage along the backbone of an RNA single-stranded molecule is similar, is the same as DNA, where the phosphate group is attached from the 5' end to the 3' end of a chain. So the phosphate linkages extend from the 5' to 3' end. So new nucleotides are added to the 3' end of a growing chain during its synthesis, and the bases stick out on the other side. Now, RNA is synthesized as a single-stranded molecule. However, once it is synthesized, it can curl up on itself and form double-stranded regions. So complementary regions of nucleotides within a single-stranded RNA molecule can hydrogen bond to each other. They can form all kinds of different structures within the RNA molecule, where non-complementary regions protrude from a region of complementarity. So you can have bulges, little loop here. You can have an internal loop where you have non-complementary nucleotides on both sides of a double-stranded region. You can have a cruciform structure like what's seen in the middle here. Right here, type of a cruciform structure. Or you can have a very iconic structure that we see in RNA molecules called a stem loop structure, where you have a region of complementarity here, and then a region of non-complementarity that loops out. This is also called a hairpin. A study that appeared in Nature a few years ago demonstrated that RNA can indeed form helical structures. So this is what a strand of DNA looks like when it has the hydrogen bonding indicated here between the complementary nucleotides. Remember, uracil takes the place of thymine, so here it is bonded to A. And if you allow the structure to take up its thermodynamically stable form inside a cell, you get a helical shape like this. And you can see that it has grooves, a major groove and a minor groove. And it can actually form variations on what's called an A form of helix, depending upon what the sequence of the nucleotides is. Now, this structure makes the RNA molecule more stable than if it was just a long, floppy, single-stranded RNA molecule. This is the form that most RNA takes up. It's called an A form. You can also see DNA take this form up too under certain salt and pH conditions. So here's our B form of DNA with a widened major groove and a minor groove. And this A form is what RNA usually forms. And it has a shallow exposed minor groove, a very narrow and deep minor groove. And if you want to look at more of this type of structure, you can go to this website. Here is a very common type of RNA molecule. Oops, let's go back. This is a tRNA molecule. And tRNA is often drawn as something that looks like this if you're looking at just its two-dimensional form. Up here is where the amino acid gets attached, and down here is the part that binds to the messenger RNA, which we will talk about during our discussion of translation. So the 3' end of the tRNA molecule sticks out here. This is where our amino acid gets attached, right here. And our 5' end is right in the molecule itself. It's right close to it. So it's one long strand of RNA. If you start at the 5' end and you go down and you follow it around and it loops around and it comes down here. This is the anticodon that hydrogen bonds to the messenger RNA. And it goes back up and around and back, and it finally gets back to the 3' end. Now, In order for tRNA to maintain a constant, stable structure, it forms these double helices, these regions of double helices. Right here, it's a very important one, and right here. This is a very beautifully rendered, three-dimensional space-filling model of a tRNA. There are many different tRNAs that accommodate the entire genetic code, but each one of the tRNAs, although the sequences are slightly different from each other, they all have the same shape so that they can fit into the same spots in the ribosome. More about that later. One other interesting fact about RNA molecules is that they can also be catalytic. That extra oxygen on the 2' carbon allows a lot of catalytic reactions to be undertaken by RNA molecules. So enzymes in a cell can be protein, that's probably the majority of them, but there are also some very, very important RNA enzymes that we will talk about later on in our course. So in summary, RNA nucleotide structure is similar to DNA nucleotide structure. It has a sugar, base, and phosphate groups. There are some very important differences between the sugar and the bases, however, between RNA and DNA. The 2' hydroxyl makes the molecule more reactive and less stable. This allows some RNAs to be catalytic, which we'll talk about later. Although it is a single-stranded molecule, it can inform internal double-stranded hairpins, all different kinds and shapes and forms and sizes. The A form helix is different from the DNA B form. Their grooves have different accessibility to protein. The helix is wider and more compact. The bases are not perpendicular to the axis. And finally, although more labile than DNA, some structures, for example, tRNAs, are very stable.